Well, thank you. As I said, it's really great symposium. And today I would like to take this opportunity and to talk about that our share of this COVID-19 studies. And our lab is focused on the lungs, and I would like to talk about findings about COVID-19 lungs, and with a focus on these proliferating cells. Next one. Okay. But before I go into the details of the phenotypes, I would like to take one step back and talk about the lung stem cells in the respiratory system, which include the trachea, and then you have these intrapulmonary airways, and then all these intrapulmonary airways become smaller and smaller. Then we're open to these millions of breathing units called alveolar, which is consist of these arteries and veins, and then also, of course, then those epitheliums, which are responsible for gas exchange. Within this system, now the stem cells or progenitor cells exist in these different locations. For example, in a human trachea and a mouse trachea, and also human intrapulmonary airways. Basal cells are the stem cells of these locations. For example, in the trachea, these basal cells can give rise to ciliated cells and club cells, and then also minor population of the mucous secreting cells, whereas you can see the basal cells sitting at the bottom part of this pseudo-stratified epithelium express markers like P63, which is transcription factor, and then you have this keratin 5 as well. And then they can give rise to these club cells marked by this FCGB1A1, and then ciliated cells can marked by acetylated tubulin, and also the transcription factor FOXJ1. And my ex-colleague Jason Rocks has shown that if you isolate these single basal cells, they can form these spheres, and if you section out through these spheres, now you can see that these single basal cells can give rise to other cell types. And if you culture in this 2D or transfer model, now these single basal cells will give rise to these red club cells labeled this FCGB1A1, and then you have this acetylated tubulin positive ciliated cells as well. But this only occurs in the humans with a trachea and intrapulmonary airways. But in mouse, which is a little bit different, the stem cells is actually called club cells. Because in a mouse, basal cells are barely detected in intrapulmonary airways. And Amarolians have shown that CC10, which is a marker, also called FCGB1A1, showing here. She made a mouse line to do lineage tracing to show that these club cells can give rise to ciliated cells, but also these club cells can self-renew to make more club cells as well. But I also want to mention is that in a mouse, these club cells is heterogeneous population, including stem cells, but also the other cell types as well. And my colleague Wellington Cardoso has shown that there's a subpopulation of club cells can has a more potential to become stem cells. Go down a little bit further into this alveoli. Now you have these type 1 and type 2 cells, and in study in mouse and also human in vitro culture have shown that these type 2 cells can serve as a stand of progenitor cells to make type 1 and type 2 cells. In this case, for example, Babcock's Christina have shown that single isolated base 82 or type 2 cells can form this sphere, and then this sphere can include the cells like more type 2 cells, but also type 1 cells marked by this protein called T1 alpha, and then another protein marked type 1 cells called aquaporin, also shown up in this single type 2 cell formed sphere, suggesting that these type 2 cells can give rise to both type 1 and type 2 cells in vitro culture. And of course, we haven't shown here is that if you do lineage tracing to label this single 82 cells, they can see now these 82 cells that can give rise to more 82 cells, but also in 81 cells under injury repair situation. And a group in UNC recently has identified that SARS-CoV-2 receptor ACE2 is enriched in ciliated cells and also type 2 cells. And as I mentioned to you, ciliated cells exist in trachea and intrapulmonary airways, whereas type 2 cells exist in the alveoli. At the very normal state, you can see that some of these cells don't proliferate, but then consistently you can see now in those COVID-19 patients, the epithelium lining the trachea and airways are severely damaged. And then in the alveoli, you can see in the damaged side, the alveoli has you can see been destroyed, but in some area now you have some air sacs being preserved. So the question is, how does this different type of stem cell respond to this injury? And it's something that we are interested in. In a normal state, you can see any proliferating cells in the airways, but also in the alveoli. And this is one case that you can see there's only a single KRTS positive basal cells exist in the airways. And blow-up region showing here, they can see blow-up region. You can see that proliferating basal cells in here, and now you have these club cells labeled by this FCGB1A1, and then you have this K5, keratin 5 labeling basal cells sitting at the bottom. But in patients with this COVID-19, now you can see extensive proliferation going on in the trachea with this labeled with keratin 5, which is basal cell marker, and then you have this red staining of these proliferating cells extensively present. And going further down into the intrapulmonary airways, now you can see, especially in those airways with a diameter larger than 0.5 millimeter, and you can see the majority of proliferating cells are basal cells there. And in very few, occasionally you can see some proliferating club cells as well. But the majority is basal cells. But when you go further down into the smaller airways, which is diameter smaller than 0.5 millimeter, then you can see the proliferating cells, they are not basal cells anymore. You still can see a few, with 30% of the basal cells proliferating. But then you can see the majority of proliferating cells shown by this Ki67 staining, they don't express markers like keratin 5, and they also don't express other differentiated markers like club cell marker, SCGB1A1 or FOXJ1. So we call this cell population called the lineage inactive proliferating cells, which we count this cell number. You can see about 30% of the basal cells and about 30% of proliferating cells are basal cells, and around 4% proliferating cells are club cells. But then the majority of proliferating cells are lineage inactive proliferating cells, and they don't express any differentiation marker. And we also look at other cell types, and for example, mucin secreting cells, which is marked by this marker 5AC. We don't see any of them proliferating, and this is in large airways or small airways. And none of these proliferating cells are marked by mark 5AC. Also, we have looked at the neuroendocrine cells as well, and we don't see any of these neuroendocrine cells which mark by none of these proliferating express none of these neuroendocrine cells express Ki67, suggesting that both mucus secreting cells and neuroendocrine cells don't proliferate. But when you go into further down into the alveoli, you can see now majority cells, proliferating cells are type 2 cells in here. In the damaged side, you can see that the small arrows, they point to those proliferating type 2 cells, and this is one of the blow-up region of this tree, and you can see the cell proliferating cells express both Ki67 and then shown by this green staining, but also you have this red staining of this type 2 cell marker, SPC. and both in this boundary region between the preserved area and the damaged area, and showing two. You can see those proliferating cells sitting at this boundary between damaged area and preserved area. In those preserved area, you do see some occasionally, proliferating type 2 cells, but very few. But contrast, you can see in type 2, in a damaged area, you see extensive proliferation type 2 cells, suggesting that they are responsible for making new type 2 cells and also differentiate into type 1 cells to trying to regenerate the lungs. So I put all this these proliferating cells in this cartoon to show you that in a COVID-19 lungs, in a trachea and in a intrapulmonary airway with diameter larger than 0.5 millimeter, the majority proliferating cells are basal cells. But in those small airways with diameter smaller than 0.5 millimeter, then the majority cells are those lineage negative proliferating cells. You do have these 30% of the proliferating cells are basal cells, and then occasionally you see some of club cells differentiate. So, and in the alveolar now, type 2 cells kind of proliferating cells, breathing. So then the immediate question that we're trying to address now is that what is the identity of those lineage negative proliferating cells? And then, of course, we would like to potentially culture those lineage negative populations and ask what these cells can give rise to. Can they make more club cells or make more basal cells as well? And hopefully that we could find a way to manipulate the differentiation of these lineage negative cells into different differentiation cell types. And with that, I would like to thank the people in this picture, but also people that are not in this picture, and a few postdocs in here. And I also want to slide our Columbia University Biobank, and especially Dr. Anjali Saki, who helped us to generate the provide the samples, and Dr. Hanina Hiboshishi, who coordinated this, help us to get those samples with low PMI. And I also like to thank Kevin in histology core to help us provide some slides as well, and my funding agency. And with that, I'm very happy to answer any questions.
So thank you for a very excellent talk.